Hi, everybody. My name is Ralph-Axel Mueller. I'm a professor of psychology at San Diego State University, and I will be talking about brain connectivity in autism. And I have a little sticky on my computer to remind you that you can answer questions online while I'm talking. And if there's enough time after my talk, I will get to them and try to answer them. So let me start off with some basic things about autism. As you surely know, it's a neurodevelopmental disorder that has a high prevalence and in fact increasing prevalence. Recent estimates are above 1%, so that's a pretty big public health issue. Autism is diagnosed in terms of behavioral criteria, which is rather crucial and a crucial problem, as I will describe in a few minutes. The most recent diagnostic criteria, at least in the US, are taken from the DSM-5 that came out last year, and it focuses on deficits in social communication and interaction, restricted and repetitive behaviors, and these symptoms need to be present early in development, and they need to be clinically significant, so relatively severe, and not explained by general intellectual disability and so on. Now, autism is thus diagnosed as a socio-communicative disorder, but it's in fact much more than that. If we look at the neuropsychological and behavioral profile, we see that numerous other cognitive and sensory motor domains are affected in ASD. And there's also an enormous individual variability and heterogeneity within the disorder. And the field has come to accept that from the biological perspective, it's probably several or maybe numerous etiological pathways that result in a phenotype that then meets the criteria for autism. So there's some kind of convergence here between biological pathways and the phenotype. People have talked about autism in the plural, and that doesn't sound very pretty. So the terminology these days is autism spectrum disorder. That's also how the DSM-5 talks about it. Another thing that we know about autism is that there's strong heritability. Going back to twin studies, we've known this for a long time. However, the problem is that hundreds of genes have been identified that are somehow involved in ASD, and it appears that each of these genes conveys only very small risk for the development of ASD. On the upside, there's been some convergence of findings suggesting that many of these risk genes have something to do with synaptogenesis, synaptic function, and circuit formation. And that obviously already brings us to the topic of my talk. But before I go there, I want to ask some a really simplistic, naive question that's nonetheless important. Why don't we just cure ASD if it's such a big public health problem? And let me raise a few crucial questions that stop us from doing so. So for fully targeted treatments that could result in a cure, we need some mechanistic model of the causation of autism during brain development. And we don't really understand those causes. We know that genetic factors are very important, but how do the hundreds of genes that have been identified trigger the etiology that leads to a phenotype that we diagnose as autistic? And then virtually everybody in the field accepts that ASD is a brain-based disorder. Then why do we diagnose children based on purely behavioral criteria? Well, the reason is that we don't know what the brain biomarkers are for ASD. And that brings me to the topic of my talk. So to get started, don't even try to read this slide. It might come out too small on your end anyway. And it's I'm not going to go through this in detail at all. This is just a segment of findings on brain anatomical abnormalities in ASD. And you can see that almost every part of the brain has some findings. So frontal lobe, temporal lobe, parietal lobe, subcortical structure, cerebellum, and so on and so forth. And I could, if I really put everything that's been found on this slide, I'd probably have to use a 3-point font or so. But the point is, there seem to be abnormalities all over the place in the brain. Now, there are some interesting and rather specific findings, though, that do affect or relate to the brain as a whole, or most parts of the brain, going back to Eric Cochen's milestone study in the early 2000s, that have to do with early brain growth. When we look at gray matter volume, this is the neurotypical growth going up, and then actually in mid-childhood starting to go down again. So cortical thickness actually goes down. That's a good thing in typical adolescence. In autism, there seems to be early overgrowth of gray matter, and then accelerated reduction of volume. And these findings overall have been supported by other groups. So that's a pretty firm finding, early overgrowth finding. What's important is that we also see this in white matter. So white matter is actually too big early in life, in the first years of life, but then white matter growth, which actually continues in typical development for quite a long time, is stunted in ASD. So connectivity is most likely abnormal from very early on in life. So now I will turn to the kind of core topic of my talk, which is functional connectivity and the search for biomarkers in network connectivity. Well, what is functional connectivity? Let's start off with a really simplistic and loose definition of functional connectivity. It's how brain regions talk to each other. At a later point, I will become a lot more specific, but the term is really very loosely defined. And the idea is that they don't talk well to each other in ASD. Let me illustrate the very, very simple basic principles of functional connectivity MRI by looking at our very first fcMRI study led by Michelle Villalobos almost 10 years ago. And what she did is she extracted a time series. Functional MRI generates time series from primary visual cortex, as illustrated there along the calcarine fissure. And then this time series is correlated with the time series of other voxels in the brain. So we see an example here of a time series in an inferior frontal voxel, and it's highly correlated in a typically developing participant, and much less correlated in a participant with ASD. And we're looking only at one example of one voxel here, but you can imagine how the whole thing works when we do this for the whole brain. And what we found in this study was indeed significantly reduced connectivity between primary visual cortex and bilateral inferior frontal cortex. And I won't go into the interpretation of these findings. I just want to say that this fit in very well with the general literature back then. And the whole thing actually got started a year earlier by Marcel Just's 2004 paper, where he looked at sentence comprehension in autism. And the activation patterns that he found are shown here, and they're not drastically different. But what his group found was that a number of brain regions showed reduced signal correlations, bold signal correlations in ASD. and he formulated an underconnectivity theory of the autistic brain that was then supported by subsequent studies, many subsequent studies from his group, and also studies from other groups. And these lists are not complete. These are examples of studies, because obviously the fcMRI literature has grown quite a lot. I can't give you completely exhaustive lists of studies here. But a lot of evidence at this point suggested that, in fact, we have a firm finding here. The autistic brain is underconnected. See this review paper by John Hughes there. However, there's a problem. There have been a lot of studies, especially recently, that are completely, or at least partially, inconsistent with the general underconnectivity hypothesis in ASD. And a disclaimer here, right away, you see some studies listed there. Again, not a complete list. About half of these are from my lab. And in fact, some of the earlier ones, like Akiko Mizuno's study in 2006, it was very hard to publish this study because back then reviewers simply did not want to believe that there could be something other than underconnectivity in autism. Let me give you some examples of studies that show different effects. So this is a study led by Mark Shen that was an activation fMRI study looking at semantic decision, and you see the activation clusters here, the biggest one in Broca's area, left inferior frontal cortex. And what we did in the functional connectivity study is to use the cluster of shared activation between both groups. We used that as a seed for functional connectivity analyses, and those are the results that we find. And in the TD group, we see relatively distinct functional connectivity findings in posterior parasylvian cortex, Wernicke's area, and then also in the homotopic inferior frontal cortex in the other hemisphere, the right hemisphere. In ASD above, the picture looks much more distributed and diffuse, and direct group comparison actually shows us that many parts of the brain are overconnected with inferior frontal cortex in ASD. So we have diffusely increased both correlations. In another study, we tried to dig a little deeper and understand how that might be related to the organization of cortex. In this study led by Patty Shee, we looked at posterior superior temporal sulcus, and we identified 3 subregions within posterior superior temporal sulcus that were functionally distinguished: a rostral region, a middle PSTS region, and a caudal region. And they're color-coded, or their connectivity patterns are color-coded here. And you can see that they're quite distinct, so we don't have much overlap between those red connectivity areas for the middle seed and parts of the brain that are connected to the caudal and rostral areas. Those two, however, show relatively similar connectivity patterns. When we look at the ASD group, we see much less differentiation. So when you look at these dark areas, those show shared connectivity for all 3 subregions in PSTS. So that is what I mean by reduced differentiation of connectivity patterns. Now, interestingly, this reduced local differentiation was associated with overconnectivity. And again, same color code here, you see regions that show significant overconnectivity in ASD. So we have 2 findings, basically. We have a reduction of local functional differentiation and an increase in long-distance connectivity. And these 2 seem to go together. They are associated. And it's expected that these, or one could assume that these are two manifestations of the same neurodevelopmental abnormality. Now, I owe you an explanation, because I've been telling you, or started off by telling you, there are all these studies that show underconnectivity in ASD, and now I showed you two studies that show the opposite. So what's going on here? So let's step back to the very first steps in fcMRI research. And that goes back to Bart Biswell's work in the mid-1990s. So what we see here is an fMRI activation result for bilateral finger tapping. And you see activations in bilateral motor cortex and supplementary motor area. And what his group did was to pick out one of these primary motor cortical activations and apply this cluster as a seed to a resting state data set. And now in the picture on the right, you see resting state, correlated resting state fluctuations in the rest of the brain. And you can see that the correlation is specifically seen within motor cortex. So there are spontaneous fluctuations within the motor network. And since then, thousands of studies have used this approach to map out various functional networks, not just the motor network. You can apply this to pretty much any network. Subsequent work has shown that the important signal correlations are found in the very low frequency domain, below 0.1 hertz. This is work by Dietmar Cordes. These are basically the interesting correlations that are driven presumably by neuronal fluctuations, whereas the rest is, the other colors indicate physiological noise. Why do we think it's neuronal activity? Some studies have shown that there's actually a link to slow fluctuations in local field potentials. And interestingly as well, these low frequency fluctuations of the bold signal can be modulated by learning and practice, and they are thought to reflect Hebbian plasticity. But most interestingly, for our purposes here, is the fact that these are fluctuations that occur spontaneously. You can find them in the absence of a task. And that brings me now back to our question in ASD and our inconsistent results. So functional connectivity results depend on design and type of analysis, and we can distinguish two broad types of analysis. One is co-activation, or I would call co-activation functional connectivity MRI. And here we're looking at correlations that are driven by a task or by stimuli. And these studies tend to focus on the effects between activated regions of interest, and they tend to find underconnectivity in ASD. On the other hand, we have an approach that's called intrinsic functional connectivity MRI, where we look at spontaneous low-frequency fluctuations. We use low bandpass filtering to isolate those fluctuations. And if we have a task or stimulus presentation, we actually remove those effects statistically, or we use the resting state. We'll come back to that later. These studies actually tend to generate overconnectivity findings in ASD. Okay, so I'm claiming that, but why would you believe me? Well, let me try to convince you. First of all, we did a literature survey a few years ago, where we looked at all the fcMRI studies that had been published in ASD up to that point, 32, and we sorted them by their findings, those that found underconnectivity only and those that found overconnectivity or partial overconnectivity. They're color-coded here. And what we found is that these two types of studies differ significantly in their methodological fingerprints that are illustrated here with this radar plot. And the significant findings were for low-pass filtering. So all studies that had shown overconnectivity had used low-pass filters, whereas only half of the studies that resulted in underconnectivity. Hardly any of the studies, of the underconnectivity studies, had used task regression, removal of task or stimulus effects, whereas most studies finding overconnectivity had done that. And a third significant finding was for field of view. Virtually none of the 21 studies that had found underconnectivity in ASD had actually looked at the entire brain to see whether there were maybe inverse effects elsewhere, whereas many studies that have found overconnectivity had looked at the entire brain. We've since then followed up on this literature survey study with our own empirical investigation using 3 data sets, 2 task data sets and 1 resting state fMRI data set, to directly compare the effects of methodological variables. So we analyze these data sets along competing pipelines. And summary of the findings is that there were significant effects for type of analysis. So co-activation analyses tend to yield underconnectivity findings in this graph up, and intrinsic fcMRI analyses tend to yield overconnectivity findings down in this graph, and a significant finding of field of view. So if you only look at regions of interest, you're very likely to find underconnectivity, but it's different when you look at the whole brain. So the crucial variables are temporal bandpass filtering to isolate low frequency fluctuations, and if there is a task, regressing out the modeled task effects. Studies that focus on high frequency task-driven effects will tend to yield underconnectivity in ASD. Studies that focus on low frequency intrinsic correlations will tend to yield overconnectivity findings in ASD. And these overconnectivity findings occur mostly outside activation ROIs. So if we only look at activation-derived ROIs, we might not see them. Now, there was another finding in this study that the data set used also had a significant effect. And I can't discuss this in detail, but I will come back to the resting state and issues with the resting state a little later. So what does all this mean for ASD? Rather than having a general principle of underconnectivity in ASD, I think that points towards a dual impairment in ASD. So if we look at a cartoon network on this brain here, so it's not supposed to be actual regions, but just kind of a simplistic illustration here. So we have integration in the TD brain between 3 nodes of this network, and this integration is reduced in ASD. However, that's not all. We also have atypically increased connectivity outside the network, so reduced differentiation between networks. These regions are not supposed to talk to each other a lot, but they still do in ASD. An example was the language network talking too much, atypically, to visual cortex in one of the studies that I showed earlier. So using a metaphor, we could say that we have impaired network sculpting in ASD. And this actually relates to the model by Mark Johnson of how networks form in typical development. And there are basically two mechanisms, or types of mechanisms: progressive mechanisms like myelination and synaptic strengthening, and regressive mechanisms like synaptic pruning and axonal loss. And obviously these are experience or activity driven. Both of these are impaired or appear to be impaired in ASD, resulting in inefficient network organization and what one could call noisy information processing. And in fact, this approach is also, model is also very consistent with some recent work out of the UCLA group, Mirella DePreto and Susan Buchheimer's group. Let me show you one more study, a recent study by Ina Fishman and who looked at mentalizing and mirror neuron systems in ASD, so theory of mind and MNS networks. And what she found was that although there was a pattern of both over and under connectivity when we looked at we might be back here. Okay. That we're back. Okay. All right, I'm sorry about that. We had some technical problems here. So I was trying to go to the next slide. So basically, I've been showing you a bunch of findings that support this network sculpting model. Before it gets too pretty, I want to bring up some problems here. Or caveats. One of them, there's probably some regional and network specificity in these principles that I described. And let me just give you one example, a study where we looked at connectivity between thalamus and cerebral cortex, and you can see the cerebral cortical regions here that we used as seeds. And we can do a parcellation of thalamus based on anatomical connectivity, color-coded in the same way. It comes out pretty similar in typically developing in ASD groups. However, there are significant differences in anatomical connectivity from DTI, which I will turn to in a little bit. We see underconnectivity, reduced connection probability between various, or for all of these cortical seeds in thalamus. And we also see in functional connectivity, MRI, reduced functional connectivity. The findings are not always exactly the same, but there is obviously quite a lot of convergence here of findings, with the exception of findings for the temporal lobe and connectivity with thalamus. Some of these findings are related to diagnostics, but not all of them. And this is not always very straightforward. And I can talk about it in the Q&A if you want to. So, complicated story. Two things. So this is an example of regional or network specificity. Here, the principles that I mentioned don't seem to apply in the same way. And this is probably also true for some cortical networks. Two added comments here. We used resting state functional connectivity MRI here and DTI. I have to talk a little bit about DTI. So let me do one after the other. Let me say a few things about the resting state. Can we assume that when we put a participant with ASD into a scanner that they will respond to an undefined condition where we just tell them, just relax, in the same way as a typically developing child? Probably not, actually. And there's actually some evidence. So let me give you one example. That's Dan Kennedy's fMRI study, where he looked at deactivations actually during task performance compared to the resting state. And these deactivations reflect activations during the resting state. And you can see these blue clusters on the left there. That's the default mode network. That is tends to be active during the resting state in typically developing adults. And you can see that in adults with ASD, that was not seen at all. So he called that failure to deactivate. So the problem is that if kids with ASD go into cognitive states that are different from those seen in TD participants during this resting state, we don't really know because we don't have any control over it, and it's very hard to monitor that. So if we have findings of underconnectivity within the default mode network in resting state fcMRI studies, we don't really know whether that's maybe just because kids with ASD don't go into this default mode. And one way to try to approach this is by looking at dynamic changes across time in functional connectivity to address the question of whether some children with ASD maybe go in and out of the default mode. And we find that in some cases. So these are some examples, and you can see that the connectivity between the default mode network stays close to ceiling in these two TD participants, and they go up and down, and the connectivity goes up and down across time in ASD participants. But these are selective examples, so this is still an ongoing study. But what Mark Mulvey in a presentation at SFN found was that regions of interest that are underconnected in ASD tend to also show increased variability across time. So that's consistent with this general idea that it's because of variability of cognitive states that we have these findings. So as promised, I want to talk a little bit about anatomical connectivity, diffusion tensor imaging being the technique of choice here. And just very briefly, how does this work? We look at water diffusion. We look at a fiber tract here, shown vertically in this cartoon. These circles and ellipses indicate water diffusion. That's what we pick up with diffusion tensor imaging. If we go outside the fiber tract, say we go into CSF, diffusion is isotropic, the same in all directions. But inside our fiber tract, it's anisotropic. So a fractional anisotropy is up inside a fiber tract. And there are other indices like mean and radial diffusion. Mean diffusion is just the overall diffusion. Also goes down within a fiber tract, and so does radial diffusion, indicated by these arrows there, also reduced in the fiber tract. So increased FA and reduced MD and RD are signs of well-organized white matter fiber tracts. Just one example of an ASD study using tract-based spatial statistics, which is a technique that allows us to map out the main fiber tracts across the brain in green here, the so-called skeleton, and then test for differences in these DTI indices across all these fiber tracts. And the red spots on these images indicate regions of significantly reduced fractional anisotropy in children and adolescents with ASD. And you can see they're found widely in many different parts of the brain, many different tracts. Another index of white matter compromise is increased mean diffusion, and again we see this kind of finding many tracts across the whole brain, and the same with increased radial diffusion. And don't worry about all these abbreviations; these are different tracts. And the take-home message here is solely that we have widespread white matter compromise, and there's largely consistent evidence in a pretty large number of studies in children, adolescents, and adults with ASD. I've cited here only a couple recent ones. There is a very recent caveat study from Nancy Conwisher's group suggesting that differences in head motion might be behind some of these FA differences. However, the study that I showed you, in that study, groups were matched for head motion. This is obviously a serious concern, but I don't think all these findings will go away completely once we learn how to address motion confounds better in TTI research. A more important caveat is that patterns in infants and toddlers are probably very different. Let me show you two examples from this so far very small literature. This is a study by Ben Bashat's group, looked at fractional anisotropy in children between ages 1.5 and 6, and you can see that in fact FA in part of the corpus callosum was increased rather than reduced in that age group. They found that also in other parts of the brain. This is just an illustration for one part of corpus callosum here, and we're looking at the same segment of corpus callosum here in another study by a consortium group, study led by Wolf and colleagues. Now, the principal finding is the same, that there is actually increased FA very early in life in ASD. However, the timing is very different in this study. So according to this study, this only is true up to maybe 12 to 15 months of age. But the conclusion at this point is the same. There is early white matter overgrowth, or these findings, these FA findings, could be related to early white matter overgrowth. It's very tempting to believe that. It also, again, drives home the fact that connectivity is quite certainly aberrant in ASD from very early on in life. Now, what I've shown you though so far have been mostly studies that focused on specific networks, specific regions of the brain, specific tracts of the brain. And I want to make the point at the end of my talk that these approaches, these hypothesis-driven approaches, might actually not be sufficient in ASD. Let me explain why. It's really very, very easy in ASD to find the rationale for just about any hypothesis. If you have an area of the brain that you really like and you really want to claim that it has something to do causally with ASD, you will find some supportive evidence in the literature. I gave you an example of MRI, anatomical MRI studies at the beginning of my talk, but that's true for many other types of techniques. We have too many findings. But on the other hand, we don't have enough strong, compelling, and replicated findings for strong hypotheses for unique biomarkers. Biomarkers may be very complex, and they may include unexpected brain patterns that at this point we have no basis of hypothesizing. And importantly as well, I talked about the probably existence of subtypes of ASD at the beginning of my talk. Now, complex biomarkers may actually be clusterings of complex brain patterns. It is very important as a first step to get an idea what these biomarkers could be so that we can then approach these questions. And all of these questions require data-driven approaches for investigation. So let me give you some really simple examples first of data-driven approaches. And the first one, study led by Ryan Cardinale, used independent component analysis and resting state functional MRI. Independent component analysis can map out functional networks in a data-driven way. And these networks are characterized by shared temporal variance. They are maximally independent. And when you run ICA on MRI data, you end up with some noise components, and you can throw them away, and then you end up with components that you have criteria for selecting as true components, functional components. And we found 13 of these functional networks that were well matched to networks known from the literature. And 10 of these showed differences in asymmetry, hemispheric asymmetry. And the interesting finding here was that all of these showed rightward asymmetry shifts in ASD. So there appears to be a persistent shift in hemispheric asymmetry in ASD. Now that's interesting, and that's almost a different topic here, but without data-driven approaches, we would not have been able to find this, because these networks are not defined by any particular hypothesis or regions of interest, but they are data-driven. Graph theory is another type of approach that allows us data-driven characterization of connectivity abnormalities in ASD. In this study led by Chris Keown in my lab, we looked at local connection density within a 14 millimeter radius, and these are the density patterns across the whole brain in TD and ASD children, and they look overall similar. You see very hot regions of high density in posterior visual cortex and also in medial prefrontal cortex. However, a direct group comparison shows us that there are significant differences in posterior cortex, primarily visual cortex, highlighted here, circled. But there are also some inverse effects of local underconnectivity in more anterior regions. The really important finding in this study was that these overconnectivity clusters in posterior cortex, or the degree of overconnectivity, was associated with symptom severity in the ASD group, suggesting that there are links between symptomatology and local connectivity. Now, probably the most important approach, data-driven approach in ASD, is diagnostic classification. If we can train an algorithm to tell us this brain is autistic, this brain is typically developing, then we're getting very close to finding those biomarkers. This is an example study by Jeff Anderson's group using a resting state functional connectivity MRI matrix, and a very large matrix with over 7,000 regions of interest, and a sample of 80 participants in a very small validation sample. One interesting finding that they made is when they looked at their connectivities by Euclidean distance here on the x-axis, they found a lot of underconnectivity. The blue cells there, these reflect underconnectivity in autism for connections that were mid to long distance. And interestingly, these tended to be the most informative connections. Informative in the sense that they were strongest, they most strongly contributed to the diagnostic classification, ASD versus TD. However, the overall prediction accuracy was only about 70% in this study, so this is still relatively low. Colleen Chen in my lab has been pushing forward these diagnostic classification efforts using machine learning. In these studies, we've actually used consortium data from the ABIDE consortium in a larger sample selected from ABIDE. These are low motion data sets, high quality data sets, resting state, 220 ROIs. From these, the machine learning algorithm selected 100 most informative features, and we reach a classification accuracy of close to 91%, with about a 9% error rate in the out-of-bag validation sample. Now let me show you those most informative features. That's a connectogram. Frontal lobe at the top, occipital lobe, the back of the brain at the bottom, subcortex at the bottom. You can see this is not very obvious. There's not a very obvious pattern. The red stuff is overconnectivity, the blue stuff is underconnectivity in ASD. That suggests that there may be very, very complex patterns of atypical connectivity that we need for diagnostic classification. When we look at this by networks, we see slightly more of a pattern. And what's interesting here is that somatosensory networks seem to be disproportionately important or informative for diagnostic classification, and that relates in interesting ways to the growing literature on sensory motor abnormalities in ASD. All right, let me come to some conclusions. So I hope I've been able to convince you that there's a lot of evidence on aberrant functional and anatomical connectivity. But when we get to the fine print or the specifics, it becomes more complicated. The conventional model of general underconnectivity definitely requires modification. An alternative model that I've characterized with using the metaphor of impaired network sculpting seems to be more adequate, describing the literature, which shows reduced integration, in other words, underconnectivity within neurotypical networks, but also reduced differentiation in ASD, diffuse overconnectivity outside neurotypical networks. Aberrant connectivity is probably present very early in the autistic brain. That is supported by findings of early white matter overgrowth, but also by some tantalizing recent findings of what appears to be precocious aspects of maturation in years 1 or 2, and maybe a little later from diffusion tensor imaging. And that, again, probably relates in interesting ways to the evidence of early sensory motor abnormalities in ASD. So why is all this important? Coming back to the questions that I raised at the beginning of the talk. As I stressed there, we need biomarkers for ASD to get away from behavioral diagnostics. And measures of network connectivity are very promising to yield such markers. Biomarkers may be very complex. I've only shown you an example from functional connectivity MRI, but of course we want to include other MRI modalities and other neuroscience techniques in this effort as well. So they may actually become even more complex. But they may allow us to identify brain-based subtypes of ASD based on clusterings of biomarkers. And why is this important? Well, identification of these brain-based subtypes opens up really important perspectives, because one would hope that for each of these biologically defined subtypes, the links with specific genetic and environmental causes may be more transparent. I'd actually like to change my slide. Maybe more tractable. Okay, transparent is probably too much of an expectation, but hopefully they'd be more tractable. And obviously, this would be a first step to a mechanistic model of what causes autism in brain development, which is a prerequisite for fully targeted treatments that will allow us to cure this disorder. All right. Thank you very much for listening. And thanks to the participants in our studies, to the funding agencies, and the many people in my lab and outside the lab that helped with these studies. And I will now go to the Q&A. And actually, I'm asked to bring up a slide here that reminds you that you can get CME credits. Click on this box and let's see. Okay, so apart from diagnostics, does the finding of poorer connectivity have any therapeutic implications? Well, I think This may have been a question that was asked earlier on. I think I just made it clear in my last slides that, in fact, the reason why we're doing this connectivity work is that we are looking for biomarkers that put us on the way to being able to identify subtypes of the disorder. And as I tried to explain, this is a crucial step to unraveling or disentangling the etiologies that lead to a phenotype that is diagnosed as ASD. Second question, is it possible that autism or ASD could be a dysfunction in the pruning period? Well, I surely think that the functional connectivity findings that I presented reflect reduced pruning in ASD. Now, I do not believe that there is one specific mechanism or parameter of brain development that can be picked out and that will explain the entire developmental causation of the disorder. So I would say that reduced pruning or impaired pruning probably plays a role, but it's really only one factor. And it plays a role, or it can account for these effects of diffuse overconnectivity, outside network overconnectivity that we see in ASD. I showed you several examples of studies that are showing this. So the question obviously is this a primarily biologically effect, maybe gene-driven, or is this actually an effect of reduced experiential plasticity? Because we know that kids with ASD don't interact with the environment in normal ways, so it's possible that reduced experiential interaction with the environment actually is a major factor in the reduction of experiential effects of pruning. Next question. Is there a possibility that ASD would be perhaps an epigenetic issue? Yes, I should have probably mentioned that when I talked about genetic factors, that in fact there's growing evidence that it's potentially an interaction between genetic factors and environmental factors. Genes that make a child's brain more susceptible to environmental toxins, for example. That was actually an oversight. I should have mentioned that earlier. So this is absolutely a possibility. Another question. We have 2 more minutes. Okay. ASD is more common in males than in females. Have there been observed differences? Let me see. Trying to open this question. In brains of male versus female. All right. So I can't see the end of the question. Oh, I see it down there. Male versus females with autism. Now the problem is that obviously there is a high ratio, male to female ratio in ASD. In high functioning ASD, this ratio kind of tends to be even higher than 4:1. So many studies include, including our studies, include only relatively few participants with, or participants that are girls or women. So in terms of the findings that I showed, at this point I cannot tell you what differences there may be between female and male functional connectivity, for example. And I don't know of any studies that have looked systematically at that. There's some studies that have looked at differences in brain anatomy, but not there's not much evidence for connectivity. Next question, should the search for cellular and molecular mechanisms also shift from hypothesis-driven towards a data-driven analysis? Well, yes, and I think that's already happening. I think, for example, a large consortium genetics studies have used data-driven bioinformatics tools. That's not my specialty, but yes, the answer is probably largely yes. However, once we have a clear idea about biomarkers, then of course we have to go back to hypothesis-driven studies. How does childhood disintegrative disorder fit in the big picture of ASD, aberrant connectivity, and biomarkers? I apologize, I don't know enough about childhood disintegrative disorder to answer that question. So perfect timing. We are out of time. So thank you very much again for your interest.